This is Cell Transport and Homeostasis Lecture Part 2. If you have not watched Part 1, please exit this and go back. Also, make sure that you have your cellular transport and homeostasis student notes because this part of the lecture will complete your notes. So we've been talking about passive and active transport. We've gone over the structure and function of the cell membrane, and we looked at diffusion and osmosis. So as a refresher, let's take a quick look at how diffusion works.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Okay, and as a comparison, let's take a look at osmosis really quickly also.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule such as urea is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Okay. So today we are really going to focus, we're going to start with osmosis and what happens when water moves in and out of cells. How does it affect the homeostasis, the balance? How does it affect the shape? How does it affect how a cell can regulate itself? Our last clip of the day. So what I want you to pay attention to is the essential question. What happens to a cell that's placed in a hypertonic, a hypotonic, and isotonic solutions? Also, this clip has two parts to it. It also has a scenario. Imagine if you were lost at sea. Should you drink the seawater if you're really, really thirsty? Why or why not in terms of what would happen to your cells and then to you? Pay attention.
Imagine that you're stranded on a raft in the middle of the ocean. You don't have any emergency food or water, but you also know that humans can only survive about 3 days without fluids. Should you drink seawater or not? Stick with us. We're going to find out and also review hypertonic, hypotonic, and isotonic solutions along the way. Cells have a very finite range of conditions in which they can survive. If it's too hot, too cold, too watery, too salty, too acidic, or too basic, they can't function properly. We're going to explore what happens when you place a cell in a solution that is much more salty, hypertonic, much more watery, hypotonic, or has an equal salt concentration, isotonic. Remember that cell membranes are selectively permeable. They allow certain particles to pass through, but not others. Usually, larger molecules can't fit through the membrane without special channels. The cell membrane is fitted with special protein channels called aquaporins, which allow water molecules to pass through without expending energy. However, the ease with which the water molecules can cross is going to depend on something called the concentration. Concentration is a measure of how much solute there is per volume of solvent. In other words, whether the liquid is more salty or more watery. We'd usually expect that the molecules would follow a process called diffusion. They would flow from areas of higher to lower concentration. Eventually, they will reach equilibrium, where there will be an equal concentration on both sides of the membrane. However, in the case of a liquid like salt water, the salt molecules are much, much too big to fit through the membrane, so the only particle that can move is water. Let's look a little more closely. Water molecules are highly attracted to salt molecules. They cluster around salt and really don't want to move. The positive parts of the water molecule stick to the negative parts of the sodium chloride, and vice versa. The interactions between oxygen and sodium and between hydrogen and chlorine are called ion-dipole interactions, which you might remember from chemistry. Water molecules that are not stuck to a molecule of salt are far more likely to relocate. Today we're going to immerse a cell in 3 kinds of liquids: a very solute-rich liquid, a very watery liquid, and a liquid that's in between. Then we'll see what happens to the water molecules. In this first scenario, the liquid surrounding the cell has many molecules of solute in it. This liquid is hypertonic compared to the cell. It has a much higher concentration of solute particles and a much lower concentration of water. Unfortunately, the solute molecules can't pass through the membrane to reach equilibrium, but water molecules can. The molecules of water on the outside of the cell are going to be obstructed from passing through by the many molecules of solvent. However, the molecules on the inside have much less solvent getting in the way. Water will begin rushing out of the cell. Some water will come in, but the net movement, the overall movement of the water molecules will be outwards and the cell will shrink. We call this cell contraction plasmolysis. This is also how pickles are made. Now let's immerse a cell in a much less concentrated solution, a hypotonic solution. Compared to the cell, it's much less salty. Some water molecules will exit the cell, but even more will rush in because they aren't obstructed by solute particles. The cell grows in size and it may even undergo cytolysis and burst. Placing a cell in an isotonic solution, where the concentrations inside and outside are equal, is much more pleasant. Equal amounts of water molecules continue to pass in and out of the cell. The system is in dynamic equilibrium. Particles are continuing to move, but the net movement is zero. The cell neither grows nor shrinks and is much more likely to survive. Now let's return to our seawater question. Seawater is incredibly salty, much more salty than our body's cells. Since it's such a hypertonic solution, if you immersed your cells in it, they would shrivel up and die. The real danger, however, lies in how your kidneys would react to the seawater. Your kidneys would attempt to remove the toxic levels of salt by using water pulled from your cells. Your body would actually use more water in removing the salt than was originally contained in the seawater itself. In summary, drinking seawater is a lousy idea even in extreme circumstances. You're better off drinking your own pee. So there we have it. Hypertonic, hypotonic, and isotonic solutions, as well as a couple of practical examples. If you found this video useful, I hope you'll consider subscribing to my channel and checking out some of my other videos.
Okay, enough of that. All right, so back to the question. Should we drink seawater for loss at sea? And hopefully you got that the answer is no. And the reason why is because even though it's water, what happens is that your cells are saltier. I'm putting little quotation marks around that, on the outside of the cells. And so even if you drink seawater, you will create a very salty environment outside your cells, and water will leave your cells, and you will actually become more dehydrated. So you'll be more thirsty. And then this could also just lead to problems inside your body, including system shutdown, because you are starving your cells of the water that they need. So let's take a look at these three different things a little bit more closely: hypertonic, hypotonic, and isotonic solutions. So this is going to answer questions 8 through 10 on your piece of paper. So the thing that I want you to think about when you have examples of this, look at the bottom of this slide. It says, remember, water moves to the saltier side, and saltier is in parentheses because I don't want you to just think of salt. I kind of want you to think about stuff. It could be sugar. It could be anything, okay? It's not always going to be salt. But you have to think about where there is more solute. Where is there more stuff? And that is the direction that the water is going to move to. So if it is saltier outside the cell, that's a hypertonic solution. It's saltier outside the cell. Okay, so if you put a cell in that situation, like when you drink seawater, cells are going to shrink because water is going to leave the cell and it's going to try to create an equilibrium with the surroundings. Okay, so when you see wilted lettuce, that is a hypertonic situation. Okay, and what happens with plants is that the cell membrane actually pulls away from the cell wall. The cell wall is still intact, but it will cause the plant to wilt and shrink. On the flip side, a hypotonic solution. Okay, so this means that it is saltier inside the cell. So water will come into the cell to try to reach that equilibrium. And if it's too salty inside of an animal cell that does not have a cell wall, then the cell can actually burst, and that would be cell death. If you have too many animal cells that rupture, you could cause permanent damage, injury, or even death. In a plant, you see this all the time in the grocery store. If you've ever gone to the produce section of a grocery store and you hear the thunder and lightning or whatever, and then it starts raining on the produce, this happened to me yesterday at Kroger, they are creating a hypotonic solution. Why do they do that? Well, water rushes into the plants and it makes the plants look more plump. So it makes them look better, longer. So think about this the next time you are shopping. ISO, ISO as a prefix means the same. So something in an isotonic solution means that the concentration of water and solute is the same on each side. It does not mean that nothing is moving in and out of the cell, but it means that the net movement is zero. So you're going to have some practice on this, and you also have some test questions. The test questions are going to sort of look like, you'll have some that will have just words, but you will also have some examples where you will look at a picture, and in the picture, you will have maybe a cell with a number, which tells you the percent of the solute, and then you will have a number that tells you the percent of solute outside the cell. And you will have to tell me which way the water is going. Is it coming in, out, or a net movement of zero? So practice that. Take a look at that. Okay. So you should have answered 8 through 10. So number 11, I want you to take a look at the net movement of water of the animal cell and a plant cell, and you need to tell me what kind of solution these cells are in. So remember, your choices are going to be isotonic, hypertonic, hypotonic. Those are your choices. So you can pause the video and make your selections. Hopefully, you did it this way. Hypertonic solution, movement net water out, so you get the shriveled cells. And isotonic solution would have been the ones where it was going in and out. And the hypotonic solution is going to be the solution where you have the big fat cells. So let's go back a slide. So in here, you have a net movement in and out. So this is the isotonic solution. You have a net movement out. This is the hypertonic solution. You have a net movement in. This is the one that is in a hypotonic solution. Okay. All right. We have one more kind of diffusion to talk about. We need to talk about facilitated diffusion. This is number 12 on your slides. So facilitated diffusion, this facilitated means to help. So it's still passive transport. So you still have no energy requirement. You're still moving from high to low, but you're talking about bigger molecules that just can't come across anywhere on the cell membrane. They need to come across in a special place using these special channel proteins. So you should think about it like this. Think about a room with a door. Air can get under the door, right? Because it's small and it fits. But if I shut the door and I wanted you to come in, you wouldn't be able to come through the wall. You wouldn't be able to come under the door. You need me to open the door so that you can come through. So that's what facilitated diffusion is. It allows things to come through in a special location. All right, let's see if this still works. I'm not sure it does. Let's see.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
Okay, so that is the end of passive transport. So we still have to talk about active transport. So active transport, take a look at this. You have this picture in your notes. So let's compare really quickly before we even get started. So notice here that we have the high concentration to the low concentration, right? So no energy is required. Take a look at the difference here in active transport. You see this ATP, that's a fancy way of saying energy. Notice that you only have two triangles here, all right? So there's more here. So it's going to go from a low area to a high area. So those are the things that you need to look for when you want to know if something is active transport. So let's take a look at active transport. This is number 13 on your notes. So again, anything that is an active transport, it's going to move from an area of low to high, and it requires energy. You might ask, why would we want to do this? Well, sometimes your body needs things that are rare, not commonly found in an environment. Some of the minerals that you need, like potassium and sodium, are rare in the environment in your body outside the cells, but your cells need it in a higher concentration inside the cell. And so you are going to use energy to transport those items across the cell membrane. Okay, so I do want you to answer this question. How is this different from facilitated diffusion? So remember the things you want to think about. You want to think about the concentration gradient, and you also want to think about the requirement for energy. So our last slide. So there are some particular kinds of active transport, and remember in the first lecture, the very first long video, they went over each one of these. So endocytosis, endo, E-N-D-O, as a prefix means to enter. Cyto refers to a cell. So endocytosis literally means entering the cell. There are two kinds of endocytosis. The first, phagocytosis, means cell eating, and these are for big, solid type things. Pinocytosis, pino means small, and this is typically small gases and liquids. Exocytosis. Exo sounds a lot like exit, and that's exactly what it means. And cyto means cell, so exiting the cell. The processes are the same, but they just happen in reverse. So the way that we do this is where vesicles come in, okay? Remember those organelles called vesicles, like lysosomes or vacuoles? So things like lysosomes are actually made up portion of the plasma membrane. So look at how this happens. If something is coming into the cell, it basically forms this pocket and it pinches off. And so surrounding these items are actually like mini plasma cell membranes. They have the waterproofing. So then they're going to have to go somewhere where enzymes can break down that cell membrane. In exocytosis, things like waste, so carbon dioxide, or generated toxic, yucky stuff that's made by your body, it will be surrounded it will fuse, meet with the cell membrane, it will open up, and it will expel these things typically into the bloodstream, which will then be removed by the body through your waste systems. Okay, so I just want to point out, I'm going to put this on my eClass page, Fin Academy review clips. There is a series of clips here that will help you get a better understanding of the pieces of the things that we've talked about. So you should have finished your notes. Make sure that you do so that you know what is happening in lab tomorrow. That's it.